All right, well, welcome back, everybody. We're moving right along here. You can see we jumped a few chapters. We're chapter 15, part 1 for this. And if you're keeping score at home, that's pages 515 to 532 in the textbook. We're going to talk about organelles, really, and how proteins get into the organelles that they belong in. This has the potential to be a long one. We'll see how long this one runs. Actually, let me make note of the time now so I have a sense when we're done how long it took. And a lot of complexity. So don't be shy to let me know what you'd like to review in class, and we'll certainly do that. But we kind of hit a new gear here in the second half of the semester, and you're going to see a change in the density and complexity of the content, I think, in what we start going over today. So our outline for today is we will talk about the organelles in a little bit more detail than we have so far, getting deep in the cell biology portion of this course. That'll include the endoplasmic reticuli, smooth and rough, and all that springs from each. We'll talk about protein sorting and the role that the Golgi body plays in that. That'll lead us to a conversation on signaling sequences on proteins. Those are like address labels on proteins that destine them and target them for their final location in the cell or outside the cell. And we will close with a number of different stories of how proteins get into different organelles. How do proteins get into the nucleus? How do proteins get into mitochondria? How do proteins get into the ER itself, etc.? So every part of a healthy cell contributes to that cell's ability to be alive. A cell, as we've even seen so far, can be thought of as a complex orchestra of activities. That's language from the textbook, not necessarily mine, and functions all trying to hold entropy, this idea of chaos, at bay. However, just because every part of a cell contributes to life doesn't mean that all of these parts are finely coordinated. Quite the opposite. It can be pretty messy. Some reaction pathways are for making glucose, like gluconeogenesis. Others break glucose down, such as glycolysis. mRNAs are degraded in some portions of the cell, but they're synthesized, of course, in transcription in the nucleus. Molecules are being released by the cell as other molecules are being brought into the cell. All of these are examples of opposing pathways. And if these opposing pathways were allowed to happen all in the same place, cells would spend a lot of time and a lot of energy simply undoing what they've just done and doing what they've just undone. We need compartmentalization. We need designated places in the cell where glucose is made or glucose is broken down so that we don't break down the glucose we just made. We need compartments in the cell where RNA is made and RNA is degraded so we don't degrade the RNA we just made. We don't want to get caught in these destructive loops where we spend so much energy to make something anabolically, only then turn around and degrade it catabolically. We want anabolism and catabolism to be separate in all of these opposing pathways. It would not be good for life if these things were happening in the same place. A lysed, broken cell where all of that chemistry of the cell can happen in that same place, that lysate, like the homogenate that we use in lab, that's chemical chaos. Everything is intermingling. All these opposing pathways are exposed to one another. That's quickly going to become a very entropic system. For a cell to operate effectively, keep entropy at bay, and remain living, we need opposing pathways compartmentalized. That means we need organelles with lipid bilayers to keep those compartments intact, which is why we spend an entire week talking about organelles and how these opposing pathways are kept segregated. So eukaryotic cells contain membrane-bound organelles, and in fact, that's the definition. The defining feature of a eukaryotic cell is that it contains a nucleus and other membrane-bound organelles. And organelle membranes are highly specific. They're specialized. In other words, the nuclear membrane is unique and distinct from the ER membrane, which is unique and distinct from the cellular membrane. This allows each of those organelles to do their specific job, and of course, because they're all membrane bound, allows the compartmentalization of opposing pathways. These membranes and the organelles that they make possible create the enclosed compartments where enzymes can carry out biochemical reactions in the peace and quiet of no opposing enzymes around them, without the worry or the concern or the energetic inefficiency of having the work just completed by one set of enzymes outdone and opposed by another. All of the organelles are existing in the context of the cytosol, so the organelles are surrounded by the cytosol, and of course the cell is surrounded by the cellular membrane. So compartments within compartments within compartments. The most prominent organelle and the defining feature of a eukaryotic cell is the presence of a membrane-bound nucleus. It is the most important of the organelles, and the nucleus is surrounded by its own specific and specialized membrane that we call the nuclear envelope. In fact, the nuclear envelope is a double membrane. It's made up of two lipid bilayers or four total layers, and we'll talk a little bit more about that as we go through this. Because we have two layers, we can't really just rely on diffusion, facilitated or passive, to get into the nucleus because we have these two walls to get through. And so there are these very specialized and beautiful structures called nuclear pores. And to get into or out of the nucleus, the only way through for any kind of molecule of any size or charge is through a nuclear pore complex. And we'll talk quite a bit about how that transport in and out of the nucleus occurs much later on in this video. The outer membrane of the nuclear envelope is part of, continuous with, unbroken with the ER, the endoplasmic reticulum. There's no break between the outer membrane of the nucleus and the smooth and rough ER. The ER is a complex collection of intraconnected membrane tubes and stacks of these membranes. And the smooth ER is the primary site of new membrane synthesis. We talked about that quite a bit when we went over phospholipids. And the rough ER is the primary site of protein production for proteins that are going to be inside other organelles, proteins that are going to be embedded in the cell membrane, or proteins that are going to be secreted by the cell. In fact, that's what makes the rough ER rough. The rough ER appears rough because it is coated in ribosomes, and those ribosomes are actively making those kinds of proteins. The smooth ER is smooth. It appears smooth in an electron microscope because it doesn't have ribosomes on it. The reason why the smooth ER doesn't have ribosomes on it is because the smooth ER is responsible for making new membranes, making new phospholipids, among other things. We're going to talk about the rough ER quite a bit later on in this video as well. But again, for now, what's making the rough ER rough is that it is studded with ribosomes that are stuck to its cytosolic face, the face of the ER membrane that is in contact with the cytosol. And the reason why ribosomes are there is because proteins are being actively made by those ribosomes, and those proteins are being fed into the interior of the ER. Those proteins are going through the ER membrane. So here's like a little cross-section snapshot of the rough ER. Here's the lumen or the interior of the rough ER. This blue thread is the protein that's in the process of being made and then folded into its tertiary shape. What's making that protein? Of course, a ribosome is, but because that protein's being woven or threaded into the ER lumen, that ribosome is stuck and recruited to the outer surface of the ER, and it's making that ER look rough, look punctuated because of the ribosomes that are studding it. The proteins that are made at the rough ER can either be proteins that are secreted from the cell or shipped to other organelles, or membrane-bound proteins, proteins that are embedded in a membrane. And in order to be embedded in, let's say, the cell membrane, those proteins are first embedded in the ER membrane. And we'll talk about how that happens as well later on. For proteins that are destined for secretion or going to exist wholly and entirely in other organelles, they are threaded completely into the rough ER lumen, and then they fold up in that lumen, as we see in this diagram here. In addition to making phospholipids, the smooth ER also makes many of the steroid hormones, testosterone, estrogen, progesterone. It's a detoxifier of the cell. Some people will refer to the smooth ER as the cell's liver, right? The lysosome is the cell's stomach, smooth ER is the cell's liver. And intracellular calcium is stored and reserved in the smooth ER as well. And we'll tap into that smooth ER calcium store later on when we talk about cellular signaling. So again, to emphasize the fact that this is one continuous unbroken structure, we can start our cursor here at the outer envelope of the nuclear envelope, right? The outer layer of the nuclear membrane. And I'm never going to take my pen off the paper. Here we've become the rough ER studded with ribosomes. We're continuous, we're continuous, we're continuous. And then we are unbroken and connected to the smooth ER down here, because remember, the ERs again, are flattened stacks of membrane tubes. And so the smooth ER is connected to the rough ER, and the rough ER is connected to the nuclear envelope. This is one continuous structure. The Golgi apparatus, or Golgi body, is distinct and completely separate from the ERs and the nuclear membrane. but it is another set of membrane tubes and stacks. The Golgi is usually found near the nucleus, although it is not connected to it, and so the Golgi is typically found close to the center of the cell. Golgi's job is protein processing and shipping. The Golgi accepts proteins and lipids that were made from the ER, from the rough and the smooth ER respectively. Usually these components arrive to the Golgi shipped in vesicles, and that's what we'll talk about in our very next lecture, what an amazing process vesicle formation and shipping is. But for now, these membrane components, protein and lipids, are made in vesicles, shipped to the Golgi. The Golgi modifies those, turns them into glycoproteins and glycolipids, sorts them to their final destination, and then ships them to their final destination in yet new vesicles. And that sorting and that shipping is much of what we'll talk about in the next video. Lysosomes, again, are the stomach of the cell. Lysosomes have a very acidic pH, a very low pH. They're involved in digesting engulfed material, but also in breaking down and recycling faulty cellular components. So proteins that are no longer functioning properly, and in fact, even entire organelles, like mitochondria and chloroplasts, are engulfed by lysosomes and digested and degraded so that their individual monomers and parts can be recycled and used to make new molecules. Prior to making it to the lysosomes, however, vesicles containing these things that are destined to be digested pass through a series of other organelle-like structures that are called endosomes. And endosomes prepare these materials for digestion. Endosomes reduce the pH in steps, for example. So endosomes will get the pH from 7 to 6, and then another endosome from 6 to 5, working our way down to the very acidic pH of the lysosomes. The endosomes also sort the contents of the vesicle, making sure that things are digestible. If there's material in the vesicle that can't be digested in the lysosome, it's going to be sent back to the cell membrane to simply be dumped into the extracellular space. So waste will be routed outside the cell, digestible material will be routed towards a lysosome, and so there's a lot of monitoring and sorting that occurs through these endosomes as molecules make their way to either the lysosome or recycled back out. So table 15.1 in your textbook just lists the organelles and a brief description of their main function. You know all this. You've been probably hearing about this stuff for a while, but it's there for you if you want to refer to it. We're going to skip any in-depth discussion on the mitochondria and chloroplast. Lord knows we went plenty into the mitochondria in the first half of the course. Right? We talked all about cellular respiration. That's purely mitochondrial function. And we're not going to talk about photosynthesis in this class. You'll get that from other classes if and when you need it for your curriculum. So we're going to focus more on the nucleus, the nuclear membrane specifically, I should say, and then the ER and the Golgi. Most of these organelles are held in place in some regard. There's flexibility and fluidity, but they're held largely in place within the interior of the cell through connections in the cytoskeleton. Cytoskeleton is also something we'll be talking about later on in this semester. And most of these interactions are occurring specifically through the microtubule component of the cytoskeleton. Organelles make up over 50% of a typical cell's volume, and you can tell what's important to a cell by how much of its volume it gives to different organelles. So if we look here, we see that the cytosol makes up about 54% of the cell volume. Okay, fine. Making up nearly a quarter of the cell volume is mitochondria. There's 1,700 mitochondria per cell, and that's taking up a quarter of the whole cell's volume. That makes sense because without ATP, what's even the point, right? We need all that ATP to bribe the universe, to allow ourselves to be this totally ordered system in a highly entropic universe. So need that mitochondria to do the cellular respiration we talked about, pump out ATP by the bucketful through the turning and churning of ATP synthase. We need power for life to be possible. Second most after the cytosol and the mitochondria, the ER. We got to make membrane, we got to make protein. 12% of the cellular volume is for the endoplasmic reticuli. The nucleus makes up 6%, which is actually pretty stunning, consider that there's only one nucleus, and it's making up 6% of the total cell volume. And then we see the Golgi at 3% and the individual other organelles at 1%. And make a note of how many peroxisomes, lysosomes, and endosomes there are per cell. So even with 400, 300, and 200 respectively, that's making up only 1% of the cell volume. That means those organelles are much, much smaller than the others. Total area of the membrane found within a single cell is enormous. The amount of membrane in a cell is huge. The ER membrane alone usually makes up 20 to 30 times the amount of membrane of the cell membrane. Remember, this is a surface area to volume issue. So the ER membrane is highly articulated. It's tubes, it's folded, it's stacked. So you can squeeze a lot more membrane into space. We talked about that with the inner mitochondrial membrane, right? It's all folded up to increase surface area. So there's a lot of membrane in the cell because a lot of the chemistry of life happens at membranes. With respect to size and overall mass, cell membrane is actually pretty tiny and puny because it's not folded and articulated. The cellular membrane is largely two-dimensional, and so what you see is what you get. It's these folded internal membranes where you get a lot more surface area and a lot more membrane. So to truly understand the relationship between organelles and the larger cell, how organelles function independently, how organelles function in a coordinated way, to even understand their evolution, we have to understand membranes, which is why we spend so much time on membrane-related processes here in the second half of the semester. So with regards to that, how did membranes evolve? How did organelles evolve? And where did eukaryotic cells come from? Well, that's a really interesting story. There's two main ways that the organelles evolved. One of those stories we've already told. It's the origin story of the mitochondria and the chloroplasts. I'm not going to repeat it here. I mean, I'll show an image in just a second, but I'm not going to repeat it here. These were engulfed prokaryotes that were not digested. They lived symbiotically with the larger cell that engulfed them, yada, yada, yada. But that's just true for mitochondria and chloroplasts. All the other organelles evolved differently. The evolutionary precursor of eukaryotic cells are, of course, prokaryotic cells, or things that were proto-prokaryotic cells, ancestors of the modern-day prokaryotic cell. In other words, cells with only one membrane, the plasma membrane, the cellular membrane, with no internal compartments whatsoever. It is believed that in this early cell, the plasma membrane had to carry the load of all cellular chemistry, of all biochemistry. Again, the chemistry of life happens at the membrane. And if a cell only has one membrane, it's the cell membrane, then all of the membrane-based processes must happen at that cellular membrane. And in fact, this is what we see in modern-day bacteria. Modern-day bacteria are counting on their cellular membrane to do all of the membrane-specific biochemistry needed for life to be possible. Now, for prokaryotes, that's okay because they're so small and they're so simple. They don't really need that much biochemistry, and so a relatively small membrane gets the job done. But as eukaryotic cells or proto-eukaryotic cells, way back in history, grew larger and grew more complex, they needed more membrane to support that complexity. Again, membranes support the biochemistry of life. And so if the biochemistry of life is becoming more complex, then you need more surface area of membrane to support that. More membrane was needed. It appears as though, because more membrane was needed, but the overall volume of the cell couldn't get much bigger, that the membranes that now make up the nucleus and the membranes that make up the ER and the Golgi and the lysosomes and the endosomes, in fact, the membranes that make up everything other than the mitochondria and the chloroplasts that were engulfed, all arose from the cellular membrane needing to get bigger, but not having the volume to grow. So if the membrane has to get bigger, but the cell can't, only thing that can happen is that the membrane invaginates, it pinches in. So if you look at the volume of this cell, and you look at the volume of this cell, and you look at the volume of this cell, you see that those overall volumes are pretty much all the same. The cell is the same size. But the only membrane we see here is the cellular membrane. Here we've invaginated, so this is all membrane gain. Look at all this extra membrane we've got now by invaginating or pinching in this membrane. And now we've not only pinched in, we've pinched off. So now we've got all this cellular membrane inside the cell now. So we believe this is how all of the organelles of the endomembrane system evolved. That the membrane of the lysosomes, the membrane of the endosomes, the membrane of the peroxisomes, the membrane of the ER, the membrane of the Golgi, the membranes, because it's two, of the nuclear envelope, all were cellular membrane that pinched in to surround and compartmentalize regions of opposing pathways. And in fact, these membranes remain connected even now. The nuclear membrane, the smooth ER membrane, and the rough ER membrane are still physically connected because they all came from the cellular membrane. And biochemically speaking, there's really no stark difference between the membranes of the endosomes and the lysosomes and the peroxisomes and the cell membrane as well. With the exception of the nucleus, because of that double membrane, all of these other organelles, all the organelles of the endomembrane system, rough ER, smooth ER, lysosomes, peroxisomes, endosomes, Golgi, they all still transfer materials with one another through vesicles. So these membranes are completely compatible. The ER can send things to the Golgi through vesicles. Vesicles are just membrane bubbles. The Golgi can send things to the lysosome through vesicles. The reason why vesicles connect all these organelles is because it's all the same membrane. And so vesicles can fuse readily with all of these different organelles because they are all derived from the cellular membrane. These membranes, again, remain compatible because they have a common source, the cellular membrane. So if we go back to the previous diagram here, you can see how the inside of these organelles are actually extracellular. So if we go here, right, see how we pinched in? Everything that's white is extracellular. When we pinch these membranes in, we trap extracellular area inside the organelle, in between the membranes that we've invaginated. And so the inside of organelles has the same pH, the same characteristics as the extracellular environment. We see this even now today. So this supports the endomembrane hypothesis that current organelles are really just cellular membranes turned inside out or invaginated from the outside world. Further supporting the idea of the endomembrane hypothesis is that we witness right now modern day bacteria sometimes, rarely, but sometimes invaginating their membranes in just this way. Specifically, we see this in the cells that make bacteriorhodopsin, for example. Bacterial cells that have these photosensitive proteins, they typically seclude those photosensitive pigments like retinol from the rest of the cell by doing these invaginations. And the result of this is kind of like a primitive almost organelle or proto-organelle. The other evolutionary origin of organelle membranes is the one we've already discussed, this one, the endosymbiotic hypothesis, where we've engulfed a prokaryotic cell and it became mitochondria. We did the same and it was a chloroplast, but again, we've talked about that before. Just highlighting now that these are not endomembrane evolutions. These organelles are different and privileged. The consequence of these two different origin stories is just as vesicles can easily and compatibly transfer materials from the ER to the Golgi, from the Golgi to the lysosome, from the lysosome to the peroxisome, vesicles connect all of those organelles together because they are all of the same membrane. Vesicles do not and cannot deliver things to the mitochondria or the chloroplasts, or vesicles don't leave mitochondria and chloroplasts either. Mitochondria and chloroplasts are other. And so vesicles are not a feasible way to get things to those organelles. We'll talk about how we do get things into those organelles in just a second. So when proteins are made in the rough ER specifically, or even in the cytoplasm, the making of the protein is really just step one. Those proteins have to be folded properly. Chaperones help us do that. We talked about that already in this course. Those proteins need to be sorted to determine where they're going, and then they need to be sent or trafficked to that area. They need to be imported by some organelles, exported by other organelles like the Golgi or the ER, and they need to get to where they're going. What gets lost also in the glamour of mitosis is that organelles have to be distributed as well. In mitosis, we talk all about chromosomes being distributed. That's great, right? I proudly prefer milk and tea as the distribution of chromosome copies. But the organelles also have to be distributed among daughter cells. Cells have no mechanism to make new organelles from scratch. Cells can't like be missing peroxisomes and just make a peroxisome. All existing organelles had to come from the mother cell. Those organelles can be grown and then distributed and divided among daughter cells when a cell separates, when a cell divides, but you have to be starting with some of them. So organelles behave in very much the same way as cell theory does, right? Cell theory states that all existing cells must have derived from previously existing cells. All existing organelles must also derive from previously existing organelles. Organelle growth requires new membrane lipids from the smooth ER. That allows the organelle to grow. Organelles can divide when they grow to a certain size, and then these organelles, because they're present in large number, can then be evenly distributed between two daughter cells. Now, of course, during mitosis, the nuclear envelope dissolves completely. So when the nuclear envelope reforms at the end of telophase, you form two nuclear envelope, and the ERs, the rough ER and the smooth ER, are positioned right at the metaphasic plate. So when the cell splits in two, the ERs are split in two, making sure that the daughter cells get half of each one. So we've got some loose threads here. We've got some complications. We've got this endomembrane system with different organelles with different purposes, proteins made in the rough ER, lipids made in the smooth ER, both sent to the Golgi for processing and then shipped to their final destination. Okay. We've got proteins that also need to get into the mitochondria or get into the chloroplast, but they can't go through vesicles because those aren't endomembrane organelles. Okay. So what this really sounds like to me is getting newly synthesized proteins to their correct destinations is a critical process that needs a lot of regulation and it's required for cell growth and cell survival. So how does all that happen? How do we sort proteins and traffic proteins to get them to where they need to be? Proteins are imported into organelles by three primary strategies or mechanisms. Proteins are delivered directly from the cytoplasm, cytosol, to the nucleus. So nuclear proteins, proteins that are destined for the nucleus, are made by free ribosomes in the cytoplasm. Mitochondrial and chloroplast proteins, same deal. They're made in the cytoplasm by free ribosomes, and they're shipped directly to the mitochondria or chloroplasts. Those are, again, the other organelles, the organelles of not the endomembrane system, mitochondria and chloroplasts. For the other organelles that are part of the endomembrane system, those proteins and lipids that are needed for those organelles to grow are going to be made by the ER, and then they're going to be shipped to their organelle destination through vesicles, through membrane bubbles. Proteins enter the ER from the cytosol. In fact, proteins are made and threaded directly into the ER. Those not destined to exist and do their job in the ER are then going to be shipped to the Golgi for further processing. If the protein is meant to be a Golgi protein, then it'll stay in the Golgi. If not, then it's going to be shipped from the Golgi to its final destination. So we have almost like this Henry Ford assembly line where the protein starts being made in the cytoplasm, but if it's an endomembrane protein, a secreted protein, or a membrane-bound protein, it's going to then be threaded into the rough ER as it's being made. If it's an ER protein, it's going to stay in the rough ER and do its job. If it's a Golgi protein, it's going to be shipped from the rough ER to the Golgi and then stay in the Golgi and do its job. And if it's a lysosomal protein or a peroxisomal protein or a membrane-bound protein or a protein destined for a secretion, it's going to be shipped from the Golgi to its final destination. Proteins made by free ribosomes in the cytosol, regardless of their destination, are going to be synthesized to contain a specific sequence of amino acids called a sorting signal or a signaling sequence. We can use those two terms interchangeably. A sorting signal is, again, a specific sequence of amino acids, usually on the beginning, the amino terminal end of the protein, but sometimes found embedded in the protein. And these sorting signals, these signal sequences, are the address label for that protein. It targets that protein to its destination. Once the protein gets to the destination that its sorting signal dictates, that sorting signal is usually snipped off and removed. And then the protein can fully fold into its tertiary or quaternary shape and do its job. So there's a nuclear sorting signal. There's a lysosomal sorting signal. There's a mitochondrial sorting signal. Each destination for a specific organelle or destination, secreted proteins have their own sorting signal. Each destination for the protein has a unique sequence of amino acids that targets that protein to that destination. This is how proteins are sorted and shipped. Proteins that lack a sorting signal, and there are proteins that are made without one, those proteins default into the cytoplasm. So cytoplasmic proteins, proteins that should be made in the cytoplasm, should stay in the cytoplasm and do their job in the cytoplasm, are the only proteins that are made with no sorting signal. Regardless of the final destination for a particular protein, a single challenge is constant between all of them, which is how to get that protein across a membrane. Well, a bunch of different strategies here as well. For the nucleus, we got to go through that nuclear pore complex that I mentioned earlier. That's the only way to get in and out of the nucleus is to get through the nuclear pore. So that's one strategy that's specific to the nucleus. Second strategy are protein transporters. Remember, a transporter is a membrane-bound protein that serves as a doorway or portal across the membrane, and it has moving parts. So we have protein transporters in the ER. We have protein transporters in the mitochondria, the chloroplasts. Proteins with moving parts, transmembrane proteins with moving parts, whose job it is to thread and transport a protein into those organelles. So that's your second strategy, protein transporters. And then the third strategy, I'm getting ahead of myself. For protein transporters, because these are fairly narrow channels, essentially, channels with moving parts, the only way that a protein can be moved through a transporter is that it has to be unfolded into its primary structure. It has to be just a straight chain of amino acids. So proteins can get into the nucleus fully folded. Proteins have to be unfolded and threaded through protein transporters. The third strategy I've mentioned already, which is vesicle transport. So proteins can be shipped from the ER to the Golgi, for example, from the Golgi to a lysosome. Proteins can be shipped to the cell membrane for secretion or to be embedded in the cell membrane, all through vesicles. And that's the third strategy, protein transport through vesicles. And all of our next video is dedicated to that third strategy. It's awesome, complex, but so remarkable and beautiful. So 3 different ways that proteins can get into an organelle. Well, fully folded protein gets through the nuclear pore complex to get into the nucleus. That's specific for the nucleus because of its double membrane. We have protein transporters, where proteins are unfolded and threaded through a protein transporter to get into an organelle. That's what we do for the ER, for the chloroplast, and for the mitochondria. Or we can ship proteins throughout the endomembrane system as cargo within vesicles. If a protein is in a vesicle, once that protein reaches its destination, the vesicle membrane is going to fuse with the target organelle membrane, and that's just going to dump the contents of the vesicle, in other words, the proteins themselves, into that destination. Yeah. So we're going to talk about the nuclear pore complex and the protein transporters for the rest of this video. And then, like I said, we'll talk about vesicle transport entirely in the next video. So a typical sorting signal, and again, we can also use the term a signal sequence, we can use those terms interchangeably, is usually somewhere between 15 and 60 amino acids long. These signaling sequences are specific and they're potent. The term we use in the sciences is necessary and sufficient, which means if you want to get a protein to the lysosome, it has to have a lysosome signal sequence on it. If that protein has a lysosome signal sequence on it, that's all it needs to get to the lysosome. So necessary, to get to the lysosome, you need a lysosomal signaling sequence, and sufficient. That's all you need. You don't need anything else. That's just going to do it. So that's what you need for correct protein trafficking. We figured out most of these signaling sequences. We know what they are, and here are some examples. The address label for getting into the ER is on your amino terminal end. Your sequence is methionine, methionine, serine, phenylalanine, valine, serine, and you can read the rest. In order to get into the nucleus, your signal sequence is proline, proline, lysine, 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 arginine, lysine, valine. Like that's the address label for the nucleus. You have that sequence in your protein, and you're going to get to the nucleus. It's necessary to get into the nucleus, and it's sufficient. Pretty cool, right? So these are these address labels, these signaling sequences that get proteins to where they're going. We've shown this experimentally in a number of different ways. We can put these signaling sequences on a whole bunch of testable, seeable, monitorable proteins, and we can see those test proteins get to the destinations due to nothing more than the addition of the appropriate signaling sequence. What we think is happening here is it's not so much the the sequence of the amino acids, right? That's how we perceive it, but it's the chemistry of the amino acids, the combination of hydrophobicity, hydrophilicity, charge, the size of the side chains, the spatial three-dimensional chemical characteristics of the signaling sequence is what allows it to be trafficked to its destination. We just see that as a sequence of amino acids. That's how we interpret. Okay, so let's talk about the nuclear pore first. Again, the nucleus is made up of a double membrane. There's four bilayers, and those two membranes are quite different even from one another. The inner membrane is often connected to a type of nuclear cortex. Remember, the cell cortex is that protein meshwork that underlies the cell membrane, spectrin in red blood cells, actin in most cells. Well, there's a nuclear cortex that gives the nucleus its structural integrity. So the inner membrane is often connected to that. And in fact, the DNA chromosomes are also associated with that cortex and the inner membrane. And the outer membrane is very similar to the ER membrane because they're contiguous with one another, right? The outer membrane becomes the ER. So we've got a stark difference in these two membranes of the nucleus. The nuclear pore is a gate that goes completely through both of those membranes. And again, I can't stress it enough. The only way in and out of the nucleus is through the nuclear pore complex. Because if you look at it, even passive diffusion doesn't get you anywhere, right? If you passively diffuse through the outer membrane, you wind up in this limbo land between the two membranes. Well, that doesn't help you. If you passively diffuse out of the nucleus, same thing. So the only way to get completely through both membranes and in or out of the nucleus is through that nuclear pore complex. So when we have a nuclear protein, a protein that's destined to exist in the nucleus, it has a job to do in the nucleus. It has the nuclear localization sequence. And let's give this some real context, right? What's an example of a nuclear protein? Well, RNA polymerase is a nuclear protein required for transcription. If you remember background from DNA replication, either from genetics or from even Bio 107, DNA helicase is the protein that separates the strands of DNA for DNA replication. We need that in the nucleus, right? We need DNA polymerase in the nucleus. We need primase in the nucleus. So all of these are proteins whose jobs are in the nucleus. These proteins have to be in the nucleus to do their jobs. These proteins are born in the cytoplasm. They're made by free ribosomes in the cytoplasm, and then they need to get into the nucleus through the nuclear pore complex. What are some examples of things that are made in the nucleus that need to get out? Well, messenger RNAs is the best example. Transcription is completely nuclear. So messenger RNAs are made in the nucleus, but translation happens in the cytoplasm. So we got to get those mRNAs out of the nucleus. Well, they got to go through the nuclear pore complex as well. In fact, the ability for a messenger RNA to leave the nucleus is one of the quality control points we have for messenger RNAs. Only messenger RNAs that were made correctly and are whole are allowed to leave. So nuclear pores are no joke, man. They're beautiful, they're complicated, and they're critical for life. Regulating flux in and out of the nucleus is a core property of life. So let's see what these nuclear pores look like and how they work. So here we see the nuclear pore complex in two views. We've got the actual view in context of the double membrane, right? So here's the double membrane of the nucleus. And then we see a nuclear pore complex in cross section. We see these fibrils, these protein fibers that emerge on the cytosolic side of the nucleus. And then we've got this basket, this unstructured protein basket, just like a net or a mesh that's on the intranuclear side, inside the nucleus. You can see these nuclear pore complexes on an electron micrograph. We're looking now at the surface of the nucleus, at the cytosolic face of the nucleus. They look like little flowers. Beautiful, right? So structured, so beautiful. Small hydrophilic things like water and ions, they can go right through the nuclear pore complex without any regulation. There's free flow of small molecules in and out of the nucleus. This is all for large polymers is what's being regulated here. The larger molecules, like RNA, like DNA, like proteins, they get caught and entangled in this basket, in this nuclear basket, this unstructured region here, catches these things like, like a net catches a butterfly. Think of it like so. These molecules, these larger molecules, in order to get through the nuclear pore complex without being caught, require a nuclear localization sequence. That's the signal sequence for the nucleus. We saw that on a previous slide on the table. If a molecule, if a protein has a nuclear localization sequence, then that nuclear localization sequence is going to be bound by another protein, a protein that's typically in the cytoplasm, cytosol, called a nuclear transport receptor. So a nuclear transport receptor is its own tertiary folded protein that recognizes and binds to the nuclear localization sequence on a second protein. And then the NTR, the nuclear transport receptor, acts as an escort. The nuclear transport receptor can interact with these fibrils and start pulling them in. When you pull these fibrils in, you stretch out this basket. It no longer becomes a net. That's the open sesame of the nuclear pore complex. And now the nuclear transport receptor and the nuclear protein that it's escorting can diffuse through the nuclear pore complex. So the nuclear transport receptor is the shuttle, is the escort that binds to a nuclear protein, and that binding happens at the nuclear localization sequence. The receptor can interact with the nuclear pore complex and essentially open the door, and then they float through together. So let's see that graphically. Here's our nuclear protein. It was just made in the cytosol. Because it's a nuclear protein, it has a nuclear localization sequence. That's part of its amino acid chain. That nuclear localization sequence is going to be recognized and bound by a nuclear transport receptor. We see that here in blue. And so now the two are partnered up. But the nuclear transport receptor, it knows how to interact with these fibrils and open the basket on the other side. So that's what the nuclear transport receptor does. Once the nuclear transport receptor opens the door of the nuclear pore complex, both of them can float through together. And then once in the nucleus, the two proteins dissociate. The nuclear protein is delivered. It's in the nucleus to do its job. If it's DNA helicase, it can help replication. If it's primase, it can make an RNA primer on a new strand of DNA. It does its job. And now the nuclear transport receptor, now empty, cycles right back out into the cytoplasm to escort in the next nuclear protein. So that's the story of transport into the nucleus. Transport into the mitochondria and the chloroplast is a little bit more straightforward. We've already told you the core parts of this. These organelles, they have multiple membranes, right? We didn't go much into the chloroplast, but we talked about mitochondria quite a bit. There's an inner mitochondrial membrane and an outer mitochondrial membrane. And so we have to get proteins into the mitochondrial matrix, right? We got to get proteins all the way into the mitochondria. So again, let's give this some context. Let's tie some things together. What's a good example of a mitochondrial protein that needs to be in the matrix? Pyruvate dehydrogenase complex is, right? The PDC, the enzyme that converts pyruvate into acetyl, that belongs in there. Ooh, our old friend succinate dehydrogenase, that needs to be embedded in the inner mitochondrial membrane because it's complex 2 of the ETC. That's got to get into the mitochondria. But some other enzymes too, like citrate synthase, the protein or the enzyme that's responsible for condensing oxaloacetate and acetyl together to make citrate. That's not membrane bound, but it's got to be in the mitochondrial matrix. So all these proteins need to be delivered into the mitochondria to do their job. So yes, these organelles, they have their own genome. They synthesize many of their own proteins, but they don't synthesize others. And those proteins need to be made by ribosomes in the cytoplasm and then transported into these organelles. So how? Well, these organelles, mitochondria and chloroplast, they have specific locations internally where the two membranes of the organelles meet. And by two membranes, I mean the outer membrane and the inner membrane. And it's almost like there's a half channel on the outer membrane, a half channel on the inner membrane, and these membranes connect to form a full transporter, a full channel. And it's through that channel that the proteins are threaded through into the matrix. So again, if we take a look at that graphically, We see here, here's a channel, right? But we don't have a connection of the membranes here. So there's no way this protein can be threaded into the matrix of this mitochondrium, mitochondrion, whatever the singular is. But here we see the half channels have come together. Here's the half channel on the outer membrane, here's the half channel on the inner membrane, and they have connected to make a full channel. Now the protein with the correct signal sequence, because it has a mitochondrial signal sequence on its end, it can associate with these channels and be threaded through in an unfolded state. Once the protein is completely threaded through into the mitochondrial matrix, two things will happen. First, the signaling sequence will be clipped off, and then the protein will fully fold into its tertiary or quaternary shape and go off and do its job. And that's pretty much it for transport into the mitochondria or the chloroplasts. Again, we have the right signaling sequence on this protein. That's why this is happening at all. That signaling sequence is being recognized by a receptor protein threaded through the correct transporter. And then once inside that organelle, that signaling sequence can be snipped off and the protein can be folded. If the protein needs help folding, there could be chaperone proteins in the matrix to help the protein fold properly, but the protein will fold and do its job. So unlike transport into the nucleus, transport into the mitochondria or the chloroplasts require the protein to unfold and be threaded through in its primary sequence, in its primary shape. Yeah, and then new membrane components that need to be transported into the mitochondria of the chloroplast. Again, we can't use vesicles to deliver those membrane components, those lipids. They're going to be brought in through specialized carrying proteins. They're going to be carried into these mitochondrial matrices, but we won't talk too much about that pathway. It's even more complex. So wrapping us up for today is the intricate and amazing story of how proteins get into the ER. And that's going to be pretty much what we close today out with. Unlike all the organelles we've discussed so far, the endoplasmic reticulum serves as the origin of proteins that are on their way to other places. Proteins that are going to be intra-organelle for organelles of the intermembrane system, proteins that are going to be embedded in a membrane, or proteins that are going to be secreted are made in the ER. They're born in the ER, and then they're shipped to other places. Proteins headed for the Golgi, proteins headed for endosomes, proteins headed for lysosomes. Again, proteins for secretion, membrane-bound proteins at the surface, and even, of course, ER proteins that are to be existing in the ER to do jobs in the ER. They're all going to be born in the ER first, and then ultimately shipped to their final destination, if their final destination is something other than the endoplasmic reticulum. Once born in the lumen of the ER, internally in the ER, these proteins will never be in the cytosol again. They'll always be ferried from organelle to organelle, from place to place, within transport vesicles. Hydrophilic, free-roaming proteins can enter the ER lumen completely, fold in the ER lumen, and exist in the ER lumen, but transmembrane proteins are also made in the ER, and they will first become bound to the ER membrane, and then that membrane will be made into a vesicle and shipped to other organelles or the cell membrane, where those membrane proteins will continue to be membrane bound. So for membrane-bound proteins, these proteins are born into the ER membrane, and then they never leave membrane. They just stay in the membrane, and they're shipped as vesicle membranes from place to place. What's causing these proteins to be made in the ER and end up in their final destination? Hopefully it's no surprise by now. It's signaling sequences. These proteins will start with an ER signaling sequence that's going to be found at the amino terminus, the beginning of the newly made protein, and that's going to direct this protein to the ER to begin its journey. Also, unlike any of the proteins we've talked about so far, proteins that enter the ER are threaded into the ER while in the process of being translated. So before the protein is completely made, transport into the ER begins. Translation continues. That protein is threaded into the ER as it is being made, and then the protein is dumped into or delivered to the lumen. So proteins that are made in the rough ER, proteins that are destined for the lumen of the rough ER or the membrane of the rough ER, the very beginning of their synthesis actually occurs with free ribosomes in the cytosol. However, because the ER signaling sequence is on the amino terminus, that's the first set of sequences, that's the first set of amino acids that emerges out of the ribosome. So once that ER signaling surface emerges from the rear end of the ribosome, that targets that protein for the ER, and all of the rest of translation occurs at the ER. The ribosome still chugging away at translation, threads that protein right into the ER lumen. And since translation is occurring at the surface of the ER and the protein being made is being thread into the ER, the protein that's in the process of being made is what's tethering the ribosome to the ER membrane, making that ER membrane appear rough. So the rough ER is rough because all of those studs of ribosomes that we see on the rough ER are ribosomes actively engaged in the translation of ER proteins. So let's see that in pictures. Right, here's a messenger RNA in blue. Here's a ribosome in green, and this ribosome has just started. It's begun its translation of this messenger RNA, and we see the new protein being made in dark green here. Well, what's the first thing that comes out of this ribosome? What's the very beginning of this protein? It's an ER signaling sequence. As soon as that ER signaling sequence emerges from the ribosome, that ribosome is going to be recruited directly to the ER membrane. How? You'll see in just a second. But it's going to be recruited to the ER membrane. Once recruited to the ER membrane, the rest of the protein is going to be made, threaded into the rough ER. That protein is going to be made into and through a ER protein transporter. That means that the new protein that's being made is the connection between the ER membrane and the ribosome. The ribosome, again, is tethered to the rough ER because of this protein that it's making. And now if you look at this ER and electron microscope, it looks rough because of all of these ribosomes that are studding it. So this is what makes the rough ER look rough. These active ribosomes that are in the process of translation, threading newly made proteins into the ER. There are no ribosomes that are permanently stuck to the ER membrane. There are no ribosomes that are permanently associated with the ER surface. All of the ribosomes, all ribosomes in general, begin as free ribosomes in the cytoplasm. If that ribosome happens to make a protein with an ER signaling sequence, only then will it be recruited to the surface of the ER, tethered to the ER by the protein it's making, and make that ER look rough. Once the protein is done, completely made, and completely delivered into the ER lumen, there's nothing tethering that ribosome to the ER membrane anymore, and it floats away back into the cytoplasm, and it's a free ribosome once again. So let's drill down on how this recruitment to the ER and this threading occurs. The ER signaling sequence, that first 6 to 15 amino acids that's on the amino terminal end of this protein, is recognized and bound by an SRP, a signal recognition particle. So what the nuclear transport receptor did for the nuclear localization sequence is what the signal recognition particle does for an ER signaling sequence. It's a protein that recognizes and binds to an ER signaling sequence when that ER signaling sequence emerges from a ribosome. So again, here's our messenger RNA in blue. Here's our ribosome in light green. Here's our newly made protein in green, but the very first amino acids of our newly made protein was the ER signaling sequence. And as soon as that ER signaling sequence emerges from the ribosome, it's going to be bound by an SRP. It's going to be bound by a signaling recognition particle, a protein whose job it is to bind to that signaling sequence. The SRP interacts with the signaling sequence on the newly made protein. It's also simultaneously capable of interacting with an SRP receptor. SRP receptors are membrane-bound proteins on the surface of the ER membrane. So now, step 2. Here's our ribosome in light green, our messenger RNA, our protein. Here's our signal recognition particle. It's binding to our signaling sequence, but it can also simultaneously bind to an SRP receptor, and the SRP receptor is anchored in the ER membrane. So now we've connected this newly made protein, which is nowhere near done, to the ER membrane. The binding between the signal sequence and the SRP slows the rate of translation down, and this gives the SRP time to find and interact with the ER-bound receptor. Once it's bound to the ER receptor, a transfer occurs. the SRP is released, and the amino acid chain of the newly made protein, the protein that's in the process of being made, is now handed off to the translocator, to the transport protein, the protein that's going to thread this protein into the ER. So that's step 3. Here's the SRP. It leaves now because it's done its delivery. And instead of being bound to a receptor, we see that we have this protein translocation channel. The signal sequence is bound to that channel. The channel actually binds to the signal sequence, and the rest of the channel is allowing the protein to be threaded through. So the signal sequence is needed first to attract the SRP. The SRP is what allows this protein to be recruited to the surface of the ER through its SRP receptor. The signaling sequence is also what allows this protein to bind to the protein translocation channel. And once bound to the protein translocation channel, this new protein will be threaded into the ER lumen as it's made. Perfect example of a protein to a protein to a protein process, right? This stepwise handoff of one protein to another to allow this process to occur. So the ER signaling sequence on the protein that's being made is what allows it to interact with and open the translocation channel. And the ER signaling sequence remains bound to that channel even while translation continues. That's why we see this looping of this protein synthesis, because the signal sequence is stuck to the channel itself. So the protein is threaded in, as we have the signaling sequence stuck to the channel. Once the protein is done, and just to remind you of Bio 107 or genetics, that protein is done when we hit a stop codon. Stop codon causes the ribosome to dissociate. So once the protein is complete and the threading into the rough ER is complete, now we're going to snip off that signaling sequence, and that allows the protein to be completely in the lumen, and that protein can fold up into its tertiary shape. And then the signaling sequence can laterally diffuse throughout the ER membrane, and it will ultimately be degraded into individual amino acids. So that's how a free-floating protein gets into the ER, a protein that might be destined for secretion, or a protein that might be completely lysosomal. What about transmembrane proteins? For proteins that are bound to become transmembrane, or destined to become transmembrane, process is a little bit different. It starts off the same way. We still have a signaling sequence, we still have an SRP, still have an SRP receptor, but the transmembrane protein has in it a portion, a piece of its amino acid chain that will become embedded in the ER membrane. And that embedded part anchors the protein into the membrane. But again, the beginning starts all the same way. There's still an ER signaling sequence. Translation occurs in the same way. Somewhere in the middle of this soon-to-be transmembrane protein, there's a second specific sequence of hydrophobic amino acids that we call a stop transfer sequence. And what do stop transfer sequences do? They stop the transfer. So because the stop transfer sequence is hydrophobic, as soon as the stop transfer sequence is threaded into the hydrophilic transporter, the blue channel evicts that stop transfer sequence. It opens up and basically like blah. Like I'm a hydrophilic channel, you're a hydrophobic patch of amino acids, I reject you, I repel you. So the channel opens up and dumps that stop transfer sequence straight into the ER membrane. Well, that works because the ER membrane is hydrophobic and the stop transfer sequence is hydrophobic. So everything's good there. But as that stop transfer sequence becomes embedded and anchored in the membrane, now as we continue making protein, the ribosome has to get further and further from the ER membrane here and make the extra, well, in this case, the cytosolic domain of the protein. So we've got a domain of the protein that's inside the ER and a domain of the protein that's outside the ER, and anchoring that protein across the membrane is the stop transfer sequence. So once the stop transfer sequence enters the hydrophilic interior of the ER membrane, it's going to be evicted by that ER channel. It'll be dumped into the ER membrane itself, and it'll get stuck there. Yeah. And then the protein can laterally diffuse, right? It can diffuse up, diffuse down, diffuse left, diffuse right. It's subject to all of what we've talked about for cell membranes in general. Signaling sequence will be cleaved as before, signaling sequence will be degraded as before, and we've got ourselves a transmembrane protein. This protein can be packaged into a vesicle and shipped to ultimately the cell membrane. And then instead of being trapped in the ER membrane, it'll be trapped in the cell membrane, which is exactly where it's needed. Sometimes the ER signaling sequence itself is not on the amino terminal end of the protein, but it's in the middle of the protein. If that's the case, we refer to that as a start transfer sequence. Here, the ribosome makes quite a bit of the protein freely in the cytoplasm. But then somewhere in the middle of the protein sequence, somewhere in the middle of the chain of amino acids, is a start transfer sequence. Start transfer sequence is essentially a signaling sequence, but it's not at the amino terminal end. When that start transfer sequence is encountered, it's bound by an SRP. The SRP brings it to the SRP receptor. SRP receptor hands that off to the translocation channel. Translocation channel holds on to that start transfer sequence, but we've already made a lot of this protein. So that becomes the cytosolic domain, a very large and articulated cytosolic domain. But then we continue making the protein. We thread more and more of that protein into the ER lumen. And if we happen to, in this case, somewhere along the way, translate a stop transfer sequence, now we've got two transmembrane spans, right? We've got two hydrophobic regions that are embedded in the membrane. So these proteins with start and stop transfer sequences are two-pass proteins. They've got two transmembrane domains. This strategy is typically used for proteins where you need multiple regions embedded in the membrane and highly complex domains on either face of the membrane. We can also have proteins that have multiple start and stop transfer sequences. And that leads to a protein that's literally threaded through the membrane, very much like stitching up a pant leg, where you've got transmembrane domain, transmembrane domain, transmembrane domain, and you're weaving this protein in and out of the membrane with multiple intracellular and extracellular faces, or intra-ER and extra-ER faces. And when we get to cellular communication, as I've mentioned before in class, we'll talk about G protein coupled receptors, which are 7 transmembrane domains. So these are proteins that have 4 start transfer sequences and 3 stop transfer sequences, threading them through the membrane multiple times. No matter how many transmembrane domains you have, whether it's just one stop transfer sequence, a stop and a stop transfer sequence, or you're a 7 transmembrane domain protein, once a protein is embedded and anchored in the ER membrane through these hydrophobic patches, that protein has to be sent to the organelle it's meant for, or ultimately to the cell membrane, so it can function in the membrane that it's destined for. Again, even if the protein is a free protein, even if it had no stop transfer sequence and it's completely within the ER lumen, still that protein needs to get to its final destination. It has to be shipped to wherever it needs to go to do its job. That shipping has to occur in a vesicle. But how these proteins get into the correct vesicle, how those vesicles are formed, and then how those vesicles identify their correct target and fuse with that target to deliver their cargo is another phenomenal story that we will talk about in the very next video. So I promised you it'd be denser, and I think it was. Let's summarize a bit of what we talked about today. First, talking about the nuclear membrane, the nuclear envelope. The outer membrane of the nuclear envelope is continuous with and part of the ER membrane. Intracellular membranes that we see around organelles very likely evolved in 2 independent ways. The membranes of the endomembrane system, that's the nucleus, the ERs, the Golgi, the lysosome, endosomes, peroxisomes, they all arose most likely through the invagination of the cellular membrane. They all are cellular. And so vesicles can move between these and share cargo in that way. The other strategy is the endosymbiotic hypothesis. Mitochondria and chloroplasts were engulfed as living prey cells, but never digested. Regardless of how this all happened, proteins have a final destination. Proteins, when they're born, have to get to whatever organelle or destination they belong in. And how to get to that organelle is the one consistent unifying challenge of this topic. And the short answer for how that occurs is through signaling sequences. these sorting signals, these small sequences of amino acids that serve as address labels and target these proteins to their final destination. We talked about three types of sorting signals that result in three types of transport that get proteins into three different destinations. Nuclear proteins have a nuclear localization sequence that is bound by a nuclear transport receptor. The nuclear transport receptor manipulates the nuclear pore complex and escorts the fully folded nuclear protein into the nucleus, which is its final destination. That's one strategy. We talked about these half channels in the mitochondria and the chloroplast that align between the two membranes, the outer and the inner membrane of those organelles, and allow a protein to be threaded through that channel in an unfolded state. And we talked quite a bit about the complexity of getting proteins threaded into the ER. For the ER, we have a signaling sequence, an ER signal sequence on the ER protein that's bound by a signal recognition particle. The signal recognition particle also binds to the ER receptor, the SRP receptor, and so the SRP serves as the bridge between the newly made protein and the ER membrane. But remember, that protein was just started in its translation. It's still in the process of being made. And so the SRP receptor hands that signal sequence off to a translocation channel. The translocation channel holds on to that signal sequence, and as the ribosome continues to make that protein is threaded into the ER lumen. For many proteins, it's threaded entirely in. We snip off the signaling sequence, protein folds up, and now we wait to ship it to its final destination. For transmembrane proteins, you're going to have at least a stop transfer sequence, a hydrophobic region of amino acids that's going to evict you from the translocation channel and embed you in the ER membrane. And then you'll have two sides. You'll stay in the ER membrane until you're packaged into a vesicle, then you'll become part of the Golgi membrane, then you'll become part of the cellular membrane. Lots of proteins have more than one transmembrane domain, and they would need at least a start and a stop transfer sequence. They're threaded through twice the ER membrane. And then some proteins have multiple transmembrane domains and multiple start and stop transfer sequences. Regardless, though, of how many start or stop transfer sequences there are, regardless if you're membrane bound at all, once you've gotten into the ER, your journey has only started. You must next go to the Golgi for further processing, glycosylation, and sorting, and then off to your final destination. And all of that has to happen through vesicle transport, which is exactly what we talk about in the next video. So you don't need me to tell you what to do. Let me know what questions you have about this material. I imagine there are some, and we'll talk about all of that in class and clarify all your confusion before we move on to the next topic. Thanks so much for watching, and I'll see you soon.